This residual limestone test involves a gasometric procedure where we aim to measure the amount of residual carbonate present in a growing medium. The overall goal is to quantify how much residual limestone is present in a substrate, because this is important in terms of pH buffering. The principle behind this protocol is to add a strong mineral acid, HCl, into a sample of substrate and then measure the amount of CO2 gas that's evolved. This chart shows increasing lime rate in units of grams of lime per liter of substrate. Remember that 1 gram of lime per liter is the same as 1 kilogram per cubic meter. 0.6 grams per liter is the same as 1 pound per cubic yard. The graph shows that pH increases, but there's a diminishing returns type of relationship. What's actually happening is that as pH increases, the reaction rate of limestone will decrease. So in this graph, we see in purple the amount of limestone that is reacted, and the yellow shows the amount of unreacted or residual lime. Most commonly, growing media have limestones that are in a carbonate form, such as calcitic limestone or dolomitic limestone. Carbonate-based limestones, when we add an acid, will produce CO2 gas. We can capture and measure this volume of CO2 gas and relate it back to the left side of the equations above to calculate how much calcite or dolomite are present in a growing medium sample. To do this, we use the ideal gas law. In the ideal gas law, we can relate the number of moles of CO2 gas liberated based on the pressure and volume of CO2, the universal gas constant, and the temperature of our system. Now, for every 1 mole of CO2 gas that is evolved from our protocol, that's equivalent to 1 mole of carbonate. And 1 mole of CO2 then is equivalent to 100 grams of calcium carbonate. Let's work our way through the protocol. You're going to need a colored reagent solution that's going to be displaced by the CO2 gas. To do this, you'll add methyl orange into a solution and bring pH down with HCl to around pH 4 to make a decidedly pink color. You're also going to need some 6 molar HCl solution to react with the limestone sample. A barometer, a thermometer, and a stopwatch are also going to be necessary for this protocol. The growing medium sample in an Erlenmeyer flask is going to be placed on a stirrer for agitation. This method uses fairly standard equipment, with the exception of the measuring burettes. If you provide a glass manufacturer with a diagram of the system, they should be able to provide you with a burette of the type that we use for our device. You'll need Tygon tubing and a lab stand, and here you can see the constructed gasometric system. So let's work our way through the general procedure. Normally we have a 50 milliliter sample of growing medium. We add some water in there to allow for increased mixing and reaction with the HCl. We add in 25 milliliters of 6 molar HCl and close the stopcock. The HCl enters our system, reacts with the carbonate limestone, and displaces our red solution as the CO2 gas evolves. We measure off the volume of CO2 gas, and spreadsheets are provided to calculate the equivalent amount of carbonate given that volume of CO2. Now there's some different steps in terms of getting accurate numbers from the system. The system should be calibrated using reagent calcium carbonate to develop a standard curve. So there's really two steps in this. The first is simply to check whether the numbers you're getting from the system are similar to the measured amounts of lime that you put into your flask. So without any growing medium, without any substrate, you can put some different calcium carbonate amounts. We suggest 0.2 and 0.4 grams. Add the HCl, measure the evolved CO2, enter in the air pressure and the temperature at the time that you're running that trial, and one of our spreadsheets will generate a calibration curve. If your numbers are very different from the numbers in this graph, then probably indicates there's some leakage or some other source of error in the apparatus. Once the apparatus is working normally, then you can develop standard curves with different sizes of substrate sample. Now normally you'll use a 50 milliliter sample of substrate, which are the top group of cells. Usually that 50 milliliters is about the right sample size. However, if there's only a very small amount of carbonate in your sample, we would recommend that you run a second set of samples with 100 milliliters. In our experience with our setup of the lab equipment, 200 milliliters is not very practical, but it can be run through the system. So here's some details here about the two different sample sizes we'd suggest, 50 or 100 milliliter samples. If the expected residual calcium carbonate is more than 0.5 grams per liter, which is equivalent to 0.5 kilograms per cubic meter, or almost 1 pound per cubic yard, then use that 50 milliliter sample. You'll need to add 75 milliliters of water, 25 milliliters of HCl, and wait for 30 minutes. If there's only a very small amount of residual calcium carbonates that you expect in the system, use that 100 milliliter sample size. You'll need to double the amount of solutions added, but the reaction time will be the same. Now to step through the procedures, take a 50 or 100 milliliter substrate sample, transfer this sample into a 1 liter decomposition flask, add deionized water to allow mixing, attach the flask to the gasometric system, put the acid dispense burette into the decomposition flask, open the stopcock to allow the air pressure in the system to equalize with outside air pressure. Adjust the left side of the burette up or down to bring the displacement solution to the 0 mark. Add the 25 milliliters of 6 molar HCl. Close the stopcock to seal the system. Lower the right side burette to reduce the pressure within the apparatus. Now introduce the 6 molar HCl into the decomposition flask. Turn on the magnetic stirrer And after the reaction is complete, around 30 minutes, level the displacement solution on both sides by adjusting the right side burette, and read off from the left side burette the amount of evolved gas. So record the volume of CO2 evolved, the air pressure, the air temperature, and enter these data into our spreadsheet, which is the residual lime calculator spreadsheet, to determine the residual lime concentration in the substrate sample. So here is a third screen for the residual lime calculator spreadsheet. And here are some data that have been entered into the system for different substrate samples. And the system will calculate the amount of residual calcium carbonate with two methods. One is with the universal gas law, and the other is by using the standard curve that you've prepared earlier. We'd like to thank the staff and students at the University of Florida who have helped to develop this technique, Dr. Bill Argo at Blackmore Company, and Paul Nelson and his team at North Carolina State University, who've done a lot of research work on limestone. Funding was provided by American Floral Endowment and the Young Plant Research Center. The Young Plant Research Center is a group of leading media and fertilizer companies and also leading growers that specialize in propagating young plants.